So some molecules are so large, so extremely large molecules or particles are so big that there's not a protein big enough to move them. So no protein can move them. Instead, they're going to have to pass across the membrane in a different process. And these are called either exocytosis or endocytosis. So again, very large particles or molecules that cannot just be carried across or have a channel for them to move through the membrane. If it's a large material exiting the cell, then it's exocytosis. So exit is exocytosis. If it's moving in, so N, in, then it's endocytosis. And there's 3 types of endocytosis that we'll briefly talk about. So exocytosis looks a lot like something we learned about in the cell chapter, and that is secretion, right? So it's also related to the secretion process, but remember secretion was for proteins. Exocytosis can be for anything. So the basic idea is that the Golgi creates a vesicle. So remember, the job of the Golgi is sorting of materials. So the Golgi is going to decide that something needs to be sent outside of the cell. So when it decides that, it will put it into a vesicle. So this is the vesicle, there's the cargo. The vesicle will travel to the plasma membrane. Remember, membranes fuse, so the membrane of the vesicle will fuse with the plasma membrane, and the little yellow triangles will exit from the cell. So that's exocytosis. Endocytosis, remember, means stuff coming in, so N for in. And there's 3 types. So there's phagocytosis. The F-A-G here root, is a root that means to feed or food. So phagocytosis is when the membrane wraps around a large particle. So here's the large particle, and the membrane is shooting out. It's kind of folding around this food particle, and eventually it's going to fold around it and form a vesicle. So there's the food particle now engulfed by the vesicle. Well, this is now a food vacuole because it's larger than a vesicle, and then the food vacuole will fuse with the lysosome, and the lysosome will digest the food. So amoebas do this, single-celled organisms do this. Our white blood cells do something similar when they attack bacteria. Another type of endocytosis is called pinocytosis. This is when the cell takes little drinks or little sips of the extracellular fluid. So this is the fluid outside of the cell. The plasma membrane will fold in a tiny bit, and when it folds in, the liquid comes in, and whatever's in that liquid will become a vesicle, and then this can be absorbed by the cell. And so this is kind of a cool image here. This is a cell, and this is a blood vessel, and so the cell is just taking little sips of what's in the blood. The last one is receptor-mediated endocytosis. So as this name gives away, this part of it's really important. So on the cell surface, and you can see them in blue, I'll just draw them a little bit bigger. There's these molecules called receptors. That has a similarity to the word receiver because receptors receive. So they're kind of like antenna, and out in the environment, in this case, they've got them as little yellow triangles. There's these little signals or something that the cell wants. And what will happen is that these receptors or receivers will catch the little triangles. And if they catch them, and that's what we see here, then the cell will fold inward. And when it folds inward, it will form a vesicle. So here's a little closer. There's the receptors. They've got the cargo, the little yellow triangle inside of the receptors. And then it will form a vesicle. The vesicle can travel to the lysosome and release that material to the inside of the cell. So in this case, the big difference is this is a selective process. In the pinocytosis, it was just folding in and taking whatever liquid came in with it. But the receptor-mediated part is being selective. It's only picking or really enriching for the yellow triangles in this case, or whatever particular cargo the cell might have. All right, so that's a wrap on transport. Exocytosis and endocytosis let us get in our big materials, and we have passive and active transport to move smaller materials in the cell.